And that's all right. It's a great pleasure to introduce Eric Reavis. And I'll go through the whole bio officially, but also just to say, so we've known it, we met, wow, was it 20 years ago? 20 years ago, I'm not too sure. Back when I was did postdoc at Harvard, so it comes in here that basically sort of did something equivalent to an honours year in the lab that I was working at, and we put up then. And anyway, I'll do the bio, and then maybe you can. Well, I'll just then I'll explain. You're also here. All right, sorry. You can see I haven't prepared this very well. Okay, so talk, as you can see, different ways of seeing, understanding altered visual perceptions. Schizophrenia. The bio, Eric is a cognitive neuroscientist at the University of California, Los Angeles at UCLA. His research seeks to understand changes in perception, cognition, social information processing that occur in schizophrenia. Completed a pre-doctoral training at Harvard. This is I was mentioning, and then the National Institute of Mental Health, followed by a doctoral training in neuroimaging and vision science at Dartmouth College. He then moved to UCLA initially to pursue a postdoc focused on psychosis research, and later joined the UCLA faculty. He's now an assistant professor in residence with the Department of Psychiatry and Behavioural Science and the Semel Institute for Neuroscience and Human Behaviour. He also holds a secondary research appointment within the Veterans Affairs Greater Los Angeles healthcare system. But what I was going to also say is that we're lucky to have Eric here in a more permanent basis. So he's moved here with his partner and working semi-remotely, so actually physically here more than physically in UCLA. I think is that in or half and half?
At the moment, yeah, I'm here a lot of the time.
Yeah.
So my professional life is still at UCLA, but my personal life is sort of...
Yeah. So we'll try and set up sort of visitor status so you can come and be around a bit more often. But just sort of to that in your brain, so that in the future, if you want to catch up, discuss some of these things, it's great that Eric is now, we'll call him a local. Anyway, looking forward to hearing the talk.
Thank you. Yeah, thanks so much for the introduction and for the invitation to speak to you all today. It's great to be here. And I'm going to jump right in because I'm going to try to cover quite a bit of material in the next 40 to 45 minutes. So let me just get started then. Yes, okay. So first of all, I have no conflicts of interest or disclosures for this talk. And I wanted to start with just giving you sort of an overview of what I'm going to talk about today. So I'm going to present some data from two studies that we've done at UCLA that take completely different approaches to trying to understand perception in schizophrenia. The first study is one that we wrapped up a few years ago, which takes a sort of classic cognitive neuroscience approach of honing in on a very specific kind of marker of perception that we use a psychophysical task to assess, and then looking for neural correlates of that marker. So it's a very sort of standard approach. And then in the second half of the talk, I'm going to talk about some work that we're doing now, which takes sort of the opposite approach, where we're kind of trying to embrace the chaos of the actual perceptual world to a greater degree, using a movie watching fMRI paradigm to look at how the brain is processing information about audiovisual stimuli under somewhat more naturalistic conditions. So these are two studies that have very different approaches, but I think sort of touch on similar issues. And there's some themes that cut across both that I hope are going to come through over the next 40 minutes. The first of us, which you might have guessed from my title, is that I'm going to try to make the point that we each have a slightly different way of experiencing the perceptual world. And it'll come as no surprise that these individual differences in perception are linked to individual differences in the brain. And then I'm also going to try to make the point that even though these differences might be quite subtle, they may actually have more consequences than it would first appear. So with that, let me just say a few words about schizophrenia, because I know many of you here are probably not clinical researchers. As Olivia mentioned, I'm originally a cognitive neuroscientist by training, but I've been working in clinical cognitive neuroscience research for the last 10 years or so. I'm sure you've at least heard of schizophrenia, even if you don't study it. And if you know anything about the illness, you probably associated with things like hallucinations, right? That's what everybody thinks of when they think of schizophrenia. That's actually not what I'm going to be talking about today. It is true that there are these important perceptual features of the illness, but even when those sorts of clinical features are pretty well controlled with medications and treatment, there are known to be various sort of persistent, more subtle changes in perception that occur in psychosis and in schizophrenia in particular, which there's been somewhere around 40 years of research on various different sort of specific kinds of perceptual measures. There's sort of a laundry list of them here. I'm not going to talk about all of them, but just to make the point that even when people with a schizophrenia diagnosis are not actively having hallucinations or other sort of perceptual symptoms, there seem to be these subtle differences in perception. And that's kind of what I'm going to focus on today. So what there hasn't been a lot of research on until fairly recently is why these perceptual changes occur in the condition. So there's been a lot of work dating back many years showing that there are these sort of differences on average, but there hasn't been a lot of research looking at the reasons why, in other words, what the neural correlates of those changes are. And so the first study that I'm going to talk about was sort of motivated by that knowledge gap, trying to understand if there were any sort of markers in the brain that related to these markers of perception. And this was a study that involved both MRI and EEG methods, as well as a psychophysical task. So the overall question that we wanted to try to address with this study was essentially whether the structure and or functioning of the brain had any relationship to a particular test of visual target detection, which I'll talk about in a moment. This was a fairly standard cross-sectional study, so people came in once. We had actually 3 groups of participants in this project. So we had a schizophrenia group, a healthy control group, and then we had a second clinical comparison group of individuals with a bipolar disorder diagnosis. There's various reasons that that sample was included, but one of the more salient ones is that there are certain kinds of symptoms of schizophrenia and bipolar disorder that can overlap. So some people with a bipolar disorder diagnosis experience psychosis during certain parts, certain phases of the illness. And there's also thought to be some shared genetic risk factors across the 2 conditions. So these 2 groups are sort of more matched on those characteristics than the schizophrenia and healthy control groups, although the 3 groups were all matched for demographic characteristics and things like that. So we had between 70 and 80 some participants in each group. The ends vary slightly depending on the different kinds of data, because we had some missing data for certain tasks and things. But those are kind of the overall ends. And I should mention that these, as well as the other, in both of these studies that I'm going to talk about today, all of the participants in the clinical groups were stable outpatients who were generally on clinically determined doses of medication. They were not very symptomatic, and they were doing well, sort of living out in the community. So these were not very symptomatic samples. They were also older samples, so like mean ages of around 46, which is not that old, but it's older than your sort of standard uni undergraduate samples, if you're used to those kinds of studies. So again, we had all these participants they all did a backward masking task, which I'll explain in a moment, an MRI scan, and an EEG, among other things. And just to explain what backward masking is, I know some of you are probably very familiar with this. It's a task that's been around forever in the vision science literature, and it's been around for a very long time in the clinical neuroscience literature as well, as a sort of common way of assessing perception in schizophrenia and in other conditions. It's a very simple task from the participant's perspective, where essentially the goal of the participant is to just detect and report the identity of a target on each trial. There's lots of different flavors of this kind of task. In our particular version of this, there were 6 different kinds of objects that appeared on different trials, and on each trial, the task was just to name the type of object that was presented. Whether it was a bell, a clock, a cup, a fan, etc. And they said this out loud. There was an experimenter sitting in the room who would enter the responses for them. So very simple task, but it's made more difficult using this masking technique. So each target is presented for a very short period, only 8 milliseconds in this case. And then after a variable delay, a high contrast noise stimulus appears in the same spatial location as the target. And depending on the duration of the delay, the target is easier or harder to identify. So when you do this, you end up with data that look like. So these are actual data from 2 participants doing this task in this study. And you can see this sort of classic S-shaped relationship between the duration of the time between the target and mask, which is on the x-axis here, the masking ISI, and performance on the y-axis, where you can see, first of all, that when there's a very short delay between the target and mask, both of these participants are just guessing. It's a 6 alternative force choice task, so chances down here around 16%. So both participants are guessing when there's a short delay between target and mask, and when there's a long delay, both participants are at ceiling, essentially. They make 1 or 2 errors, but they're easily able to report the type of target presented. In between, there's this sort of S-shaped function that describes the relationship between masking ISI and performance. And as is common in sort of standard perceptual psychophysics studies, we quantify performance on this kind of task using, by fitting a function to the data. So this is a Weibull function, these green lines here for those of you who care. And then we quantify performance for each individual by calculating the point at which that function crosses a particular threshold. By convention, that's 50% correct. So what that means is that the lower the threshold, the better the participant did on the task, right? So you see here that subject 2's data is shifted to the left relative to subject 1, meaning that subject 2 had a lower threshold and was essentially able to recognize the target with a shorter delay between the target and mask than subject 1 was. So subject 2 did better. And we can and did calculate these thresholds for each individual. And first of all, we replicated the long-standing effect that the thresholds were higher that is worse in schizophrenia. They're not different by a lot. It's about 10 milliseconds, but it is significant, and it replicates the long-standing literature. But what's more interesting and what we wanted to capitalize in this study is that there's a lot of individual differences within each group in performance on a task like this. And so we wanted to look at a few different potential relationships between those individual differences in the perceptual task and the brain. And first of all, we looked at a few structural measures. Again, we had MRI data on all of these participants, and we had both diffusion-weighted imaging data as well as high-resolution T1 anatomical scans for each person. And so we decided to look at 3 main regions of interest to see if the structure of the brain had any sort of relationship to masking performance in the clinical group. And the first thing that we wanted to look at was the geniculostriate tract. That is the projection from the lateral geniculate nucleus of the thalamus to the striate cortex, better known as V1. This is like the classic sort of main input to the cortical visual system, although it's not the only input by any means. And surprisingly, nobody had really looked at the structure of this tract in schizophrenia, at least not in vivo in a modern study. So we wanted to see, first of all, if there were any differences in the structure of this geniculostriate tract in schizophrenia, and secondly, whether those differences might relate to perception. Because our reasoning was essentially if there's something wrong with this main input pathway, that's likely to have a lot of sort of downstream effects. And then we also were interested in the structure of cortex. And we looked at 2 main regions of interest within cortex, the retinotopic visual cortex, which I'm using as a catch-all term for V1, V2, and V3 together. We looked at them separately, but the results were very similar, so I'm just going to collapse today. and also the lateral occipital complex, which is an object selective region of cortex in the lateral occipital lobe. So to start with the white matter tract, the geniculostriate tract, using the diffusion weighted imaging data that we had on these participants, we first identified the path of this tract in each individual subject's brain using a probabilistic tractography technique. And what you can see here in this figure is on the right, I'm showing you a picture from a very old textbook, Gray's Anatomy, which shows, I can see this pointer, but shows the path of what in the older literature was known as the optic radiations, the geniculostriate tract on the right. And then on the left, this is some example data from a participant in the study of the tract as identified by the probabilistic tractography. So you can see it worked pretty well. We were able to identify this tract in each person. And after we identified which voxels belonged to this tract, we looked at a few different measures of connectivity or structural integrity of the tract. Two of them were sort of classic DTI measures, fractional anisotropy and mean diffusivity, which measure essentially the amount of diffusion of water molecules either sort of along the axonal fibers in the case of FA or in all directions for mean diffusivity. And then we also looked at the volume of the tract, essentially just how large it was in each individual's brain. And we didn't see any mean group differences. So there was no sort of mean difference across groups in any of these three structural metrics. But when we looked at potential relationships with the perceptual measures, what we found was quite surprising, at least to me, which is that in the schizophrenia group, but only in the schizophrenia group, there was this surprising relationship where the individuals who had larger optic radiations, larger geniculostriate tracts, that is, they took up more space in the brain, performed better on the backward masking task. So this is masking ISI on the x-axis here and the size of the geniculostriate tract on the y-axis. And you can see there's this fairly strong relationship between the two, which we were not really expecting. And the other thing that's surprising about it is that it was unique to the schizophrenia group. So there's no indication of any such relationship in the sample of bipolar disorder, nor in the healthy controls, which suggests to us that maybe this is something related to the pathophysiology of schizophrenia itself that could be sort of driving these associations. So moving on then to the cortical ROIs that we looked at, I used a standard functional. Whoa. Okay. Actually, I'll just go back to using this. Right. There. So yes, we used a standard functional localizer technique to identify the LOC and retinotopic cortex in each person. This is the standard rotating checkerboards for retinotopic cortex and a contrast of pictures of objects versus scrambled pictures of objects for LOC. So we identified those on the cortical surface for each individual. And then we also used FreeSurfer to parcellate the brain and to identify to calculate two main structural metrics for each of those localized ROIs. The first was surface area, so just how much space on the cortical surface each ROI occupied. I can tell you up front, we didn't find anything interesting with surface area, so I won't really talk about that today. And then the other was cortical thickness. So the distance between these red and blue lines here, once the surface is reconstructed of the high resolution chemical. So we found a couple of expected main effects for the cortical thickness. So there's a long-standing literature showing that retinotopic cortex is thinner than other parts of cortex, so we replicated that. And there's also a large literature showing that there tends to be thinner cortex overall in schizophrenia, especially in older people with the condition. And we replicated that as well. So we saw these two sort of main effects showing mean group differences. But again, we were mostly interested in the sort of individual differences in these structural metrics and whether they might relate to individual differences in the perceptual metrics. And again, we saw this sort of striking effect that was unique to the schizophrenia group. So for both retinotopic cortex and for LOC, there was this relationship such that the individuals who had thicker cortex in either of these regions performed better on the backward masking task than the individuals who had thinner cortex in those regions. But again, there was no such relationship in either of the two comparison groups, which again suggests that maybe this is something that's sort of related to the pathophysiology of schizophrenia and the fact that there are these sort of effects of cortical thinning going on in the condition. So then we also looked at the function of the brain in various different ways. I won't talk about all of them for the sake of time today, but I'll just present one really simple measure of brain functioning that turned out to be surprisingly related. So we had two fMRI measures as well, but we also had EEG on these participants. And this was sort of something we had not initially planned to do, but there's a literature actually on the frequency of alpha oscillations. And if any, I imagine some of you have done EEG before. If you have, you've certainly seen alpha. It's the sort of characteristic waveform that appears typically when people are bored or just sitting quietly with nothing to do. And you start seeing these really sort of pronounced oscillations in the 7 to 13 hertz range. They're so robust and easy to see that this was the first sort of EEG waveform that was described in the literature, and that's why it's called alpha, because people could see this on their pen traces of the EEG back in the 1920s. So it's really robust. The interesting thing about it is that although everybody shows these alpha oscillations within this range, there are actually individual differences in the rate of oscillations from person to person. And those differences tend to be pretty stable, at least during adulthood. So the test-retest reliability of the rate, that is the frequency of the alpha oscillations, is quite good over time in adults. There's differences early and late in development, but not in samples typically of the ages that we had. And this resting alpha frequency, there's a literature suggesting that it's sort of like a perceptual refresh rate almost, where there's been studies in healthy participants showing that it seems to be correlated with perceptual processing speed. And there's been one or two studies in schizophrenia suggesting that it's related to measures of cognition and attention. Nobody had really looked at whether this was related to something like backward masking in schizophrenia, and we had the data, so we decided to look at it. And by we, I mean primarily Lauren Catalano, who's a researcher who's been working on this data set. We were surprised to see a similar sort of effect here. So again, this is just a measure of how fast or slow the oscillations in the alpha band are when people are sitting doing nothing during the EEG. And again, there's this sort of similar relationship between that alpha frequency and perception and performance on the backward masking task, where people who had faster oscillations were able to do the masking task at shorter threshold durations than individuals who had slower alpha oscillations. But what you can see is different here is that this isn't unique to the schizophrenia group. So there is a significant correlation in schizophrenia, but there's also a significant relationship in the healthy controls. There's not a significant relationship in the bipolar disorder group, but there also wasn't a significant difference between that group and the other two, so I wouldn't read too much into that. But this seems to be an effect that cuts across at least the schizophrenia and control groups, which suggests to us that this is maybe something that's just sort of normal variation throughout the population that's influencing all of our sort of perceptual experiences in a way, and allow and that sort of relate to even relatively simple things like this, like performance on this object recognition backward masking task. So with that, so we found these sort of interesting relationships between the structure, structural connectivity, and functioning of the brain in schizophrenia in this study. But by the time we were done with this, we sort of had the feeling that this is interesting, but also it all sort of depends on this one very specific, very controlled task, this backward masking task, which is similar to a lot of other studies that have been done, especially in this population, where you just have like one or two measures that are really robust, but they're only capturing a really sort of narrow piece of perception, right? And so there's been very little research on sort of what perception looks like in a more real world context. And so the second study that I'm going to talk about is sort of an attempt to try to bridge that gap. And so we're using a naturalistic neuroimaging technique. This is, again, an ongoing study, so we're still collecting data on this. But we're using a movie watching fMRI technique to try to get some traction on the question of how perception might differ during more real world conditions. And I'm sure any of you who've ever done an MRI before, either as a participant or as an experimenter, know that there's no such thing as truly naturalistic perception when you're lying in a confined space, keeping still. But movie watching sort of gets us a little bit closer to the kind of rich experience that that we have when we're out in the world than these very sort of highly controlled psychophysical paradigms that that we often use in the lab. And the movie watching fMRI approach kind of grew out of this landmark paper by Reavis and colleagues back in 2004, where they basically just had people come in, do an fMRI scan while watching a movie, specifically this movie. And then they looked at various things. One of them was just how correlated brain activity was across participants over time. And this was back in the days when you could publish a major paper with 5 participants. So these are their 5 participants for a particular region, or voxel, I forget, where you can see there's 5 traces here, and they all kind of go up and down together, right? So if you do a time series correlation of these, you get a pretty high R value. And this is what's known as an inter-subject correlation. So, so this was like a really surprising finding back in 2004, that when you gave people no real task, just put them in the scanner, had them watch a movie, you would get this sort of highly synchronized brain activity, especially in these regions here, sort of along the lateral occipitotemporal cortex. And so, so this was sort of, this touched off a large set of studies since then. This was now more than 20 years ago. And a lot of that work has been in the social neuroscience literature, actually. So, so my colleague in the psychology department at UCLA, Carolyn Parkinson, in her lab, she has a social neuroscience lab that's done a lot of work in this area, where the sort of overall findings that have come out of social neuroscience is that, yes, people's brain activity tends to be similar when you have them watch a movie. But there are differences, and those differences are meaningful. And I think the most striking example of this is if you look at so you do the same kind of ISC approach, you have people watch movies, you look at the correlation in brain activity across people. But if you sort the people based on social relationships, you see these really sort of major differences where people have really similar brain activity to their friends. They have somewhat less similar brain activity to friends of their friends. And when it's people they really don't have any social connection to, they really don't have very correlated brain activity at all, which is kind of striking, I think. And more recently, they've actually done some work that's prospective, where they essentially scan first-year undergraduates who come to UCLA, who've all been assigned to live in the same dormitory on the UCLA campus. So they come in not knowing one another. They get an MRI scan at the beginning of the term, and then later on, toward the end of the academic year, the researchers come back and ask, so who's friends with whom now? Because they've all been like living together and they've kind of formed social networks. And they can actually predict pretty well from the brain activity during the MRI scan, who's going to go on to become friends with whom. So it's like having a sort of similar experience of the world seems to kind of make it easier to connect with other people is the sort of way that this is thought of in the social neuroscience literature. And I know this maybe seems like we're not talking about perception anymore, but I think maybe we are in a way. So the idea is that people sort of have different experiences of the world and of these sort of stimuli that are somewhat naturalistic. And we're able to kind of capture these global differences in the way that people process this information using this ISC technique, where it seems to kind of capture the fact that some people sort of process the stimuli differently. And the work in social neuroscience and healthy samples suggests that these individual differences in the way that we experience the world, both in a sort of metaphorical, but maybe in a somewhat literal sense as well, seem to relate to our capacity to kind of connect with one another and form social relationships with people. So Carolyn and I started talking about this a while back, and we realized that this kind of technique might actually offer some new ways of thinking about two kind of interrelated issues in schizophrenia that there hasn't been a lot of work on in the past. The first is, as I mentioned, there's been very little work trying to understand perception under more naturalistic conditions, which is largely because it's just hard to do. You know, it's easy to study something when you put somebody in a lab under very controlled conditions. It's harder to measure sort of, people's experience during their everyday lives. And secondly, there's another sort of important clinical aspect of schizophrenia that I hadn't mentioned before, which is that many people with the diagnosis report difficulties forming and maintaining social relationships, which are not very well treated by current treatment options, and they're not terribly well understood either. But as Carolyn and I started talking about this, we realized that maybe these things are actually somewhat interrelated, that if, as we know from many years of literature in schizophrenia, there tends to be this sort of change in perception that occurs in the condition. And if having a sort of different perceptual experience or perceptual cognitive experience, if you want to think of it that way, is maybe associated with how easy it is to form relationships with other people, maybe these things are actually kind of connected. And maybe doing naturalistic neuroimaging in this population could give us some new insights into both of these issues. So we decided to write a grant to look at this, and we're fortunate enough to get the grant. So we're now 4 years into a 5-year study where we're measuring these kinds of intersubject correlations using a movie watching technique in schizophrenia and in healthy controls. Again, these are sort of these are stable outpatient samples with mean ages in the mid-40s, so similar to that first study. And we basically, people do various things as part of the study, but the core of it is that we have people come in, do an fMRI scan while watching clips of movies and TV shows. And I think there's eight or nine clips. They're meant to sort of capture as many different aspects of the human experience as we can in 40 minutes. And in line with the social neuroscience literature on this, they're sort of selected to be engaging, hopefully novel to people. And some of them have elements that are a little bit ambiguous, where different people might sort of pick up on certain social cues, other people might not, or people might interpret the video in a different way. So we basically have people come in and watch the same set of clips in the same order. And just to be clear, this is not a hyperscanning study. So there's no interaction between the participants here. This is just all of the comparisons across participants are done after the fact during data analysis. So, so the way we analyze these data, and by we, most of this the data that I'm showing you today has been analyzed by Lisa Shem, who's a very talented PhD student in psychology. So first of all, we do standard pre-processing of all the data. We run everything through fMRI prep and all that. And then we parcellate the brain into a predetermined set of regions. We're using a standard 200 parcel atlas. And then for each of those parcels, we basically do a time series correlation for every possible pairing of participants. So you get an R value for each possible pair of participants for each region, right? You end up for each region with a big matrix that looks like this, where you have different kinds of pairings. So you have the within schizophrenia pairings, within control pairings, and the mixed pairings in orange, green, and brown, respectively. And if you just look at the mean effects within each of the two within group pairings, it looks like this. So, so there's controls data on the top there, schizophrenia is in the second row. And what you can see just at a glance is that, first of all, the parts of the brain where the ISCs are the highest are very similar to what was found in that original 2004 paper. So it's sort of along this occipitotemporal circuit that we're seeing the highest ISCs. But there's not a huge difference between the controls and the schizophrenia group. Everything is a little bit, the colors here are a little more muted in the schizophrenia group, which indicates slightly lower R values. But it's not like the effects are in totally different parts of the brain in that sample than in the control sample. So this kind of suggests to us that the task is working as expected, right? But this is kind of more illustrative. If we want to actually do group comparisons, things get a little bit tricky because, as you may have realized, when we do these ISCs, then we no longer have participant level data. We have this dyadic data in each of those cells. So this creates some kind of statistical complexities, which fortunately there are methods to deal with. I'm not going to go into those details, but I'm happy to talk about it later if you are interested. Basically we account for those sort of aspects of non-independence and the different number of cells of different pairing types in the analyses that we do. And there's two main kinds of contrasts or comparisons that you can do that use the control group as a reference, which tell you two slightly different things. So the first contrast is this one, so comparing the mixed dyads to the control-only dyads. And if you think about what that means, what that shows you is basically whether there's a difference in processing in schizophrenia relative to controls, right? So just sort of globally, is there a difference in the way that the schizophrenia group is responding to these naturalistic stimuli versus the controls? The other kind of contrast that you can do is this one. So comparing the schizophrenia-only dyads to the control-only dyads, which actually gives you a sort of slightly different piece of information, where this is showing more whether the processing in one group is more heterogeneous than in the other group. So in other words, whether people in one group have a sort of more unique response to the stimulus than the other group. This is sometimes known as the Anna Karenina hypothesis, where it's, after the first line of the novel, Anna Karenina, something like, every happy family is alike, but every unhappy family is unhappy in its own way. This is not about happiness, but this is kind of what this is meant to capture. It's like, is each pairing of participants sort of more dissimilar in one group versus the other? That's what that comparison is showing. So we looked at both of these, and again, we used this specialized statistical method, which, if you're interested, that's the reference there. It's Chen et al. 2017, to do these comparisons. And the results so far are looking like this. So first of all, for the question of whether there are parts of the brain where the processing is different in schizophrenia versus controls, pretty clearly, yes, even though those first maps that I showed you looked pretty similar. If you do this comparison, you can see that there are, in fact, a number of regions throughout the brain where there's a significant difference. Those are the outlined parcels there. But you can also see that the magnitude of those differences is not huge in a lot of places. So we have quite a few subjects here, which gives us pretty good power. But the actual difference itself is subtle. When we look at the other contrast of the schizophrenia only versus the control only dyads, what you can see here is that it's a smaller set of regions, but the differences are more pronounced, and especially along this sort of temporal lobe here, and we see these really sort of strong differences across the STS, or sorry, the superior temporal gyrus, that are really quite striking. So this suggests to us that there is this sort of more diversified processing of the stimuli in the schizophrenia group, which is somewhat different than what's shown in this other slide, which is just sort of showing if there's any difference overall. So I'm running low on time, so just to wrap up, hopefully this second study, I know it's not the same kind of the same way of approaching perception as I'm used to, or as we did in the first study. But I hope that seeing this gives you the sense that maybe this kind of more naturalistic, data-driven approach might offer us some new insights into perception under conditions that are more like the real world. Of course, we're sacrificing some degree of specificity by having these sort of more naturalistic stimuli. But this is where I think these two approaches kind of complement one another, because we have a lot of sort of specificity with the traditional highly controlled methods. And here we have this sort of more data-driven way of thinking about how the brain is processing audiovisual information. So I won't read all the results here since we just went over them, but basically we are seeing these differences in this interim data set. And overall, I think across these two studies, the data point to the importance of individual differences overall. So again, it seems that each of us has a slightly different way of processing perceptual information, which is something we can measure both in the lab using techniques like backward masking, and perhaps also using these movie watching fMRI techniques in a sort of more global way. And we see some evidence that these individual differences in perception are linked to the brain, which as a cognitive neuroscientist shouldn't be too surprising. What was a little bit potentially surprising is that they seem to be very multifactorial sort of relationships in schizophrenia. So it's not like there's one thing that's going wrong in the brain that's leading to these alterations in perception in schizophrenia. These data and other data that have come out in recent years suggest that there's sort of a lot of small things that are probably changing that are all contributing a little bit to this sort of diversification of perceptual experiences. And lastly, I think the ongoing study is suggesting to us that there seems to be, maybe we've been focusing too much on mean differences, especially in the clinical neuroscience literature, where when we focus just on the means, we're sort of missing out on important effects of things like variability, because the newer data sets suggest that within group variation might be very important for understanding schizophrenia, and the fact that there seems to be this sort of greater variety of perceptual experiences from what we can infer indirectly from the ISC data. So I'll just quickly mention there's various ongoing analyses that we're doing with this data set. We're still collecting data, so we haven't quite finalized any of this yet. But we're also interested in things like intersubject functional correlations, which is basically looking at the correlations across different sets of regions for these dyadic pairings. We're interested in how the specific stimulus content, which varies across the different video clips and across the time domain within the video clips, whether that might relate to the magnitude of the inter-subject correlations. And then finally, one of the main eventual goals of this study, once we have a complete data set, is to try to understand whether these ISC measures might relate to social functioning or to symptoms in a way that could potentially provide the basis for something like a biomarker of social functioning in schizophrenia, which is something that we plan to look at down the road. I will wrap up there. There's some future directions that I can talk about if people are interested, but I know we're low on time. So I just want to acknowledge the many people who have contributed to this research. There's too many people to list, but these are some of the main people who've done a lot of this work. We're also very lucky to have support from the NIH to support the studies. And just want to thank the group at UCLA and thank all of you for your attention. Thank you.
Any questions?
Yeah, so really interesting research. I wonder whether you're at all familiar with some of the work from, I think it's like Lucy Berkovich and Stan Dehaene and so on, about, so I'm not a schizophrenia person, yeah, studying or have it, but yeah, I've been reading some of that work recently, which suggests that schizophrenia relates to impaired conscious access to sensory information, and kind of, you know, impaired top-down attentional amplification of certain brain activity in certain areas and so on. So like, I mean, in light of that work, I was wondering how that might relate to some of the findings you're finding here. So like in the first study, I wonder whether you've looked at kind of any other white matter tracts, in particular ones that connect like visual areas through to the inferior frontal gyrus, or sort of like, you know, superior longitudinal fasciculus, or like, you know, inferior tracts. Because I'm not as familiar as that work as I perhaps could be, but like, I think the prediction from that work is that those tracts might be disrupted, and that could maybe explain some of the effects you find where like you have like greater volume or something like that, where that trades off with something like that potentially, I don't know. And then just quickly, some of the maps you find in the differences in the inter-subject correlations, like I noticed you do see, yeah, these differences in the inferior frontal gyrus, and also like maybe those differences in the superior temporal gyrus could be explained by like less top-down activation of those representations, like as a flow-on consequence of that.
Right. Yeah, that's a very interesting question. So, so to start with the first part about the white matter tracts. So in that set of analyses, we really only just focused in on that one tract. So that was kind of that was sort of a add-on analysis that we weren't expecting to actually work in the way that it did. So we really just focused on that one tract. We haven't looked at the other potential tracts that you mentioned. We have done some other research on attention specifically in schizophrenia, and you're right that there are changes in attention as well. And so, yeah, I think it's a very good point, and it's kind of hard to say from like the ISC data how much of that is sort of the, you know, bottom up versus top down effects, if you want to think of it that way. It's probably a bit of both would be my suspicion, that, you know, we know that there are differences in attention. There's probably also differences in like what exactly people are looking at on the screen, because they're not fixating or anything. Right. So yes, I think, you know, that's another important aspect of schizophrenia that I didn't really talk about, because, you know, it would be a much longer talk. But yes, I think it probably is very important, and eventually I think it would be nice to sort of bring that into these studies. We unfortunately didn't really have any good measures of attention in these two studies. We have some really basic, like,
yeah, because I feel like it's even more than just attention, but it's like, right, capturing that distinction between like conscious access to a sensory representation or something, or just like, I think the work suggests that some of the like, you know, subconscious level processing of that stimuli is more or less typical, but
yeah, I'm somewhat familiar with that literature. I haven't read those papers recently, so I don't want to I don't know, misrepresent it,
Okay.
but yes, I think there's, yeah, I'm not sure whether that might be, you know, part of what's going on here, but I'd be happy to talk.
Yeah. Thanks.
For your second study, I was just wondering how much of it you think is it's quite a related question to the sort of sensory, like perceptual differences versus you were saying the social video that they're having, and if they have social difficulties in social cognitive difficulties, then they might be having different processing because they're just thinking about different things from those scenarios rather than something perceptual.
Yeah, so that's a very good point. So I think it, again, is probably a little bit of both. I kind of alluded at the end to how we're looking at the content of the videos and how that might influence the ISCs. So this is a way of kind of trying to get at that question to some degree. One of the things that we've done in a very kind of exploratory way is to sort of sort the videos or look at the socialness of the clips. And within the clips, we've had like undergraduates do continuous ratings of how social the video was at any given time point. And what I can tell you, although it's very preliminary and we're still looking at those data, is that the ISCs seem to be greatest when there's a lot of social stuff going on. So that part suggests that yes, there is something about the sort of socially specific elements of the stimuli that there might be greater differences in processing. That being said, even when there's not really anything social going on during the parts of the videos where there's like no people or just one person sort of sitting quietly by themselves, we still do see differences. So it's not the best test. I think just in some cases there's some overlap. In other cases it's different regions. So I haven't looked at that in a while, so I can't tell you exactly where, but I'd be happy to look at that. So yeah, I think it's probably both, but clearly the social element matters, and that's something we're also interested in.
Really great talk. I have so many questions, but I'll just ask one. For your study looking at the thresholds and the volume of is it V1? No, the thickness and V1 and also the I think you looked at another measure for the tract where you found a correlation between the threshold and that measure V1. It appeared that the value for the people with schizophrenia was similar to the ones to the controls.
Yeah.
And I wonder whether that has to do with symptom severity. So I'm not sure if you looked at symptom severity, but could it be that the ones that are doing better and cortical sweetness, the ones that are doing better and more closely resembling the healthy controls, do they have less severity in some aspect of symptomatology?
Yeah, that's a great question. So we did look at that. So we had two main measures of symptoms in these participants. One is the BPRS, which is a measure of positive symptoms, so like hallucinations, delusions, those kinds of symptoms. And then the canes, which is a measure of negative symptoms, things like kind of apathy, flattened affect, those sorts of aspects. So we looked at whether that would account for these effects, and it didn't, basically. So on the other hand, we didn't have a lot of variation in either of those two measures of symptoms because these samples were stable outpatients who generally weren't having many symptoms. So on a range, I forget the scale of those two measures, but it's like a range of 1 to 5 or 1 to 7 or something. And most people were like in the 1 to 2 range. You only got a few people who were kind of higher, more symptomatic on either of those measures. So to make a long story short, this probably isn't the best sample to test that question in, but we tried to look at it with the samples that we had, and those analyses didn't suggest that it was better accounted for by symptoms than by symptoms.
Right. Interest of time, and we've done a very good job to finish on time, so we might wrap up now and thank Eric for a great talk.